All right. Thank you for the introduction. This work is in collaboration with Henri Schmidt, who is a co-first author in this paper, Michelle Chan from Princeton, and my postdoc advisor, Ben Raphael. All right, so as you all know, all the cells in our body are related to each other. We all start as a single cell, which we call a zygote. And during developmental process, we go from this one cell to billions or trillions of cells. And all these cells are related to each other. And this relationship can be represented with a cell lineage tree, which shows you the successive cell divisions that occur during the developmental process. So one of the most fundamental questions of developmental biology is what is the history of the cell divisions during this process? As in, what is the cell lineage tree for all the cells? And this is not just specific to embryonic development. A similar kind of question can be asked also in cancer development. But typically, these cancer tumors also start when a single founder cell acquires some somatic mutations, and you go from one cell to millions of cells that might comprise the tumor. So this is a very difficult problem, inferring cell lineage trees for these complex systems. But some of you might be surprised to know that for this problem is actually solved for some simple organisms. So one such organism is C. elegans. So every single cell division and developmental fate of every cell of this organism is mapped during the developmental process. And this was done in the early 1980s using direct experimental observations and led to a lot of biological insights into genes that control programmed cell death and organ development, and later on led to the 2002 Nobel Prize in Physiology or Medicine. So, but the approach that was used here, using these experiments, cannot be really scaled to more complex organisms. So, for more complex organisms, you might want to use some heritable genetic markers, such as somatic mutations, to recover the cell lineages. And these are mutations that occur because of DNA copying errors during cell division. And these are heritable, so if a cell has it, all of its daughter cells will also have it. One limitation of using this kind of approach is that the resolution of the cell lineage tree you get with this approach is limited by the rate at which these somatic mutations are occurring during the developmental process. So in the last decade or so, there's been exciting developments in new technologies that completely bypass this problem, and these are the dynamic lineage tracing technologies. So here, artificial mutations are introduced into cells during the developmental process, which are then used to infer the cell lineage tree. And because you have more control over the rate at which these mutations are introduced, you get a better cell lineage tree. So today we're going to talk about one such technology, which is a CRISPR-Cas9 based lineage tracing. So here, CRISPR-Cas9 is used to introduce double-strand break in specific regions of the genome. And when the cell tries to repair these breaks, sometimes it can introduce insertions or deletions. And these are the heritable mutations we can then use to perform lineage tracing. What's important for us to know is that these mutations are irreversible. So once the target site has been altered, it cannot go back to its unedited state. And also, they are non-modifiable, because once the target site is altered, CRISPR-Cas9 is not able to act on it. And so for the rest of the experiment, that target site will not be modified. So let's see a simple example of 4 cells with a very simple cell lineage tree. And as you can see, there are mutations happening during cell division, so that at the end, you have these 4 cells with different mutational profiles. In this case, we just have 2 target sites. And an important thing I'd like you to notice is that the same mutation at the same target site can happen in multiple cells during this process. And this is known as homoplasy. So once this process is over, you can use single-cell RNA sequencing to measure the mutations in these cells. And that yields a character matrix that looks something like this. So you have columns that represent different target sites, which we call characters, and rows that represent cells. And the entries of this matrix are telling you the mutational state of the target site in the particular cell. So here I'm using 0 to represent the unedited target site, and integers 1, 2, and so on for the different mutations measured at these target sites. So our goal is to start from this character matrix and try to infer the underlying cell lineage tree that generated this character matrix. So that was just a very simple example. Here is a more real example of CRISPR-Cas9-based lineage tracing data, which I pulled from a recently published study that used this technology. So here, each column is a target site, and each row is a cell. So first thing you would notice is that there's a lot of colors in one column. So that shows that there's a large number of mutational states for any given character. You can have 50 to 100 states even for just one character. There's also high throughput data, so you can see that there's hundreds to thousands of cells in this kind of data sets. And lastly, there's also a lot of missing entries. So the white regions of this data matrix, these are missing entries, so you do not know the mutational state of the target sites for these cells. And in real data sets, you can get, with current technologies, up to 20% missing entries in these data matrices. So because of these particular challenges in real data, standard phylogenetic methods are not really suited for this kind of data for phylogeny reconstruction. And specialized methods have been introduced. And also recently, they were benchmarked in a dream challenge for lineage tracing. What we were interested in looking at is what would be the most appropriate evolutionary model that captures all the characteristic features of CRISPR-Cas9 mutations. So to talk about these evolutionary models, I'm going to introduce the concept of a state transition graph. So you have a state transition graph for each character in your data. So for a given character, the state transition graph has nodes that represent the mutational state of that character. So here you have, let's say, 4 states for a given character. And the directed edges of this graph tell you which are the state transitions that are allowed under your evolutionary model. So in this example, this is the most possibly permissive evolutionary model that allows all kinds of transitions. And this is known as the finite states model. And as you can see, this finite states model doesn't have the right properties for CRISPR-Cas9 mutations. So let's see some other evolutionary models that might have some or all of these properties. So the first model we look at is a 2-state Kamin-Sokal model. So in this model, you only have 2 states, the unedited state and the mutated state. And you're only allowed transition from the unedited to the mutated state. So these mutations are irreversible. And because there's only one possible mutated state, their irreversibility also implies non-modifiability in this case. The reason I'm talking about this model is that it's in fact been used in previous lineage tracing studies. But as we talked about earlier, real data actually has multiple states for each character, so this is perhaps not the most appropriate evolutionary model to use. There is a multi-state version of this model, which fewer people have studied or know about, which is a multi-state Kamin-Sokal model. But under this model, you're allowed to have transitions between mutated states, as you can see here, from the green state to the blue state. So the non-modifiability is not really enforced in this model. So realizing that none of the existing evolutionary models capture all the features of CRISPR-Cas9 mutations, we introduce a new model, which we call the star homoplasy model. And the state transition graph for this model is a star tree with the root given by where the root is the unedited state, and the leaves are the mutated states. And the specific structure of the star tree enforces all the features of the CRISPR-Cas9 mutations. So now that we have the right evolutionary model, how do we use this to infer the cell lineage tree from lineage tracing data? So in our work, we use the maximum parsimony framework to do this. Under this framework, you are given a weight for each edge of the state transition graph. And this weight denotes the probability of the mutation occurring. So if your mutation is more likely to occur during your experiment, then you'll have a smaller weight in your state transition graph. And your goal is, given a character matrix, to build a phylogeny where the leaves are the rows of this character matrix. And you want to minimize the total weight of this tree. And the total weight is given by the sum of the weights of the state transitions along the edges of this phylogeny. So, we use standard maximum phylogeny approaches to solve this problem. And We use standard phylogeny maximum parsimony approaches to solve this problem. I won't be able to go into details of that. But what I want to talk about today is, if you think deeply about this model, you'll realize that it doesn't really enforce any constraints on the cell lineage tree's topology. So you might want to, you might end up with a tree that has a large number of homoplacies for any given mutation. And that's not very realistic. So a useful version of our model is where we bound the number of homoplacies from above. And this is the bounded homoplasy version of our model. We call it the K-star homoplasy model. And here K is a user-defined integer, which is used for an additional constraint that says that each mutation can occur at most K times in the phylogeny. And this additional constraint, what it allows us to do is now we can characterize all character matrices that would admit a K-star homoplasy phylogeny. And we do this by drawing a connection between our model and a two-state perfect phylogeny model. So what is a two-state perfect phylogeny model? So this is a very restrictive model that only has two states, and you're only allowed to move from the unedited state to the edited state. And there's an additional constraint that a character can change state at most once in the entire phylogeny. So there's a lot of theoretical results on this model. Specifically, you can characterize all character matrices that admit this a phylogeny under this model. So we sort of piggyback on these theoretical results by drawing a connection between our model and this model. So the way we do this is by defining an operation which we call the K-star binarization. So this operation maps our character matrix to a binary matrix. And we show that if this binary matrix admits a two-state perfect phylogeny, this is necessary and sufficient condition for the original character matrix to admit a K-star homoplasy phylogeny. And this allows us to formulate an efficient integer linear program to find the most parsimonious K-star homoplasy phylogeny for lineage tracing. And we call this Startle ILP. So now we compare our methods to existing methods for lineage tracing on simulated data. We simulate with realistic missing dropout rates of 15%. We compare against Cassiopeia, which is another parsimony-based method. It was one of the top-performing methods in the DREAM challenge I talked about earlier. And we also compare against more standard phylogenetic methods like neighbor joining. And here from left to right on the x-axis is increasing size of the input data. On the left plot, I'm showing the distance from the ground truth. And the right is how parsimonious the solution is. And we see that our method generates more accurate and parsimonious trees than existing methods. Then we use Startle to analyze a recently published mouse metastatic lung adenocarcinoma dataset. So in this study, the authors used Cassiopeia to build lineage trees. And then these lineage trees were used for several downstream analysis, such as investigating the fitness of the tumor clones, the plasticity of tumor cells, and migration patterns that happen during metastasis. So the accuracy of these downstream analysis hinges on the accuracy of the lineage trees inferred by Cassiopeia. So we look at the largest data set from this study that has more than 21,000 cells across the primary tumor and the metastatic tumors. The first thing we notice is that our method is able to build a more parsimonious solution than the ones that are published in this study. And then we check if this leads to difference in the downstream analysis. So what we find is that starter trees actually also have fewer migrations between the anatomical sites for this case. So we use Machina, which is a method from 2018, to infer migrations from the published tree and the Startle tree on the left and right, respectively. And we show that our tree actually has fewer migrations, migration of far fewer tumor cells between the anatomical sites. And so it's giving a simpler explanation of the metastatic dissemination of this cancer. All right, so in the interest of time, I'll skip the conclusion slide and acknowledge my co-authors, Henri Schmidt, Dr. Michelle Chan, and Dr. Ben Raphael, and also my lab mates from Raphael Group and the funding sources. I'll leave the conclusion slide up, and if time permits, I'm happy to answer questions. Thank you.